I'm Andy Watson, Chief Marketing Officer at Raindance Technologies. I'm here in beautiful Amsterdam to hear from some scientists and see how some artists have used their creativity and their vision to create masterpieces. Rembrandt had tremendous vision in creating his masterpieces. I think you'll agree that his use of light was just spectacular. Could you tell me a little bit about your vision for the future of science and how your work's going to impact that future?
We would like to bring molecular analysis to the treatment of patients, to advance really the medication and the treatment of patients that can receive in the future. And this really falls into the category that we would like each patient to treat with the best treatment possible. And so we like to call this, and many other people do, personalized medicine.
I'm quite happy to live in that time now where we are faced with these new fancy technologies, and that really tremendously will change our way to handle biological questions, and also, of course, in the future, the medical care. I'm quite convinced that in the future everybody will get his sequence.
Rembrandt was an innovator in his use of new tools and new techniques. How are you using some of the new DNA sequencing tools in your work?
I see currently we need whole genome approaches to find out where the differences are and which ones are important. And since we simply don't know which differences are important in which disease, we have to look at the entire picture. Once we know where the important differences are, whether they are genetic or epigenetic, we then can do targeted sequencing of just these regions. And that gives us the power to go into thousands and hundreds of thousands of samples to really pin down what the individual contribution is to this particular disease that we are interested in.
But if you have to screen a large number of individuals, then at this moment sequencing of genomes and exomes is still too expensive. And of course you can put a filter in silico on the data, but we don't have the throughput to screen thousands of individuals. And that's where the targeted systems become very valuable.
I'm devoted to the identification of disease genes. And if I look back how long it took me in the past to identify one particular gene for one disease. It took decades. But now with the sequencing technology that we have in our hands today, it even might only take 2 weeks. So, and that really makes a great difference. And when the sequencing steps become faster and faster, so it even takes only a few days or even a few hours and minutes. So, and then it comes into the time frame as we need it for diagnostic purposes. This is already seen in the clinical application for cancer patients. Actually, today, cancer patients already can benefit a lot from these new sequencing approaches, so that they can get the right medication or the right treatment for their particular cancer.
Yeah. How do you think that Raindance Technologies will be able to provide new capabilities for you to enable new science and new discoveries for the future?
We all like to have multiple colors of paint or multiple tools in our toolbox. And yeah, Raindance is a great example of one of those tools that we could hopefully pick off the shelf and use in our studies. I think a lot of us are trying to sequence whole genomes, but we're also very nervous about how the coverage goes up and down. And if we have a particular disease, for example, that might have multiple genes implicated, genes with a lot of exons, we want to make sure that we can say, well, this patient was negative for all mutations in known genes. And if we see coverage go up and down, maybe we can't make that call for every single exon, and it leaves some questions open. So it's really nice to again, couple that tool that Raindance provides, which is to ensure that we have ultra-high coverage over the known or hypothesized candidate genes and pathways, and couple that with the goal to discover something new as well. But first we have to rule out if this individual might have had prior known mutations, you know, within genes or gene families.
But long-range haplotyping might be very important to identify interacting SNPs.
I think, you know, we have a couple ideas. One is we really see, you know, number one, from the panel side, we have a lot of ideas that we got from our XLMR success that we're going to take to the next level, especially with new things that are going to be coming out with RainDance. I think that's going to enable us to work on different types of panels. I think one very exciting thing is the ADME sequencing that we're developing together, and I think that's going to be a big market in itself.
So, Amsterdam and Rembrandt have provided us with some really great food for thought. I'm Andy Watson with RainDance Technologies, and we're looking forward to working with leading scientists to produce some of the world's next great scientific masterpieces.